Hi, I'm Merrick Zwarenstein. I'm a healthcare researcher in family medicine at Western University in Ontario in Canada. And I'd like to thank you for the opportunity for my Ottawa University colleague, Alison and Howie, and I to present some thoughts on how to make randomized trials more useful for decision making. I'll describe the different design correlates focusing on internal and external validity in randomized trials, because this is a basis for understanding the two different purposes for which you, as the trial maker, might design your trial. On the one hand, you may intend to assist decision makers like clinicians, policymakers, or patients as they choose between alternative interventions in some real-world situation, in which case your trial has a pragmatic purpose, or your intention is to test a hypothesis about mechanism of action in a tightly controlled laboratory-like experiment in free-living humans. That would be an explanatory purpose. Alison and Howie will then discuss how to use the Précis tool to make design choices for your trial, which align with the intended purpose that you hold, and illustrate these ideas with a large cluster randomized trial. Randomization is the source of internal validity. It guarantees within statistical limits that known and unknown confounders, as well as non-specific factors like regression to the mean and measurement error, are equally distributed or balanced between the arms and thus cancel each other out. It means that any difference in outcomes between the arms is the net treatment effect, provided, of course, that the randomized trial was well conducted, which means here that allocation was concealed and loss to follow-up was minimal and equal between arms. We call this miracle of statistical science internal validity. Internal, because this valid estimate of effect is accurate only for participants in the trial, not for those who were approached for the trial but refused, who were excluded, who were lost to follow-up, or were somewhere else, or are future patients. Internal validity is not enough on its own for the findings to be useful for real-world decision makers. But without internal validity, external validity is meaningless. Both internal and external validity are necessary for a trial to provide useful evidence for decision making. Randomization on its own does not ensure external validity. Now, external validity of the findings of a randomized trial is reliant on conscious design choices by you, the trial maker, designed to make your trial similar in all aspects: participants, both patients and providers, context, and care provided to the specific situation in which you intend the findings will be applied. The finding is true then outside the trial only to the extent that the situation in the trial and the external situation to which the results are going to be applied are similar to each other. There's no miraculous guarantee by statistical means of external validity. It's always a judgment call. The trial maker may, of course, be able to design their trial to exactly match the local situation with which they are familiar and in which they intend perhaps to implement the intervention if it should be shown in their trial to be effective. But only the external decision maker in an external site of application can judge the applicability of the trial to their situation. So your challenge as a trial maker is trying to design a useful trial, both for whatever situation you intended to be used, one with which you might be familiar, as well as making it useful for external decision makers. And your challenge is to keep the trial feasible for your situation, your trial site and setting, while resisting the traditional urge to constrain the design in ways which reduce the applicability of your trial for external users of your findings. Now, internal validity is dependent on randomization. External validity, on the other hand, is related to several other separate design choices that permit or constrain similarity between the randomized trial participants, the setting in which the trial is done and the care that's provided, and that of the situation to which situations, I should say, to which the findings will be applied. Since these trial design characteristics for external validity are completely separate from and fully permit randomization, there is no trade-off between them. We can both randomize to ensure internal validity and also maximize, to the extent that we would like, our external validity. Traditional default design choices that we may make that constrain the setting, the participants, and the care delivery so much that the trial may be different from usual care anywhere, even in the same setting as the randomized trial itself. And of course, this undermines external validity of the traditional approach to randomized trials and limits their usefulness for decision-making. Now, these distortions include very narrow selection criteria for patients, clinicians, and settings. Patients, for example, may be more severely ill than the usual patient in a setting like the one in which the trial is intended to be implemented, but not maximally ill. And the reason for choosing patients in this sweet spot is because they're then likely to be responsive to treatment. These patients may be selected to be highly adherent in order to avoid a dilution of effect due to non-adherence. They may be within a narrow age range in order to ensure that there's a high risk of outcome events. And they may be selected to have no comorbidities to prevent effect dilution. The clinicians and sites, on the other hand, may be selected so that there are high volume sites, which make it efficient to collect, to recruit patients and conduct the trial. And there may even be experienced sites with better outcomes for that intervention because of their experience than is the case in the usual care setting in which that intervention will be later used. Another traditional design distortion is to alter what would become the usual approach to delivery of care during the trial. That is, it would become the usual care should it be shown to be effective in this trial. For example, the trial may add extra resources to the clinical care setting. Now, these extra resources may be used to optimize the intervention, to add co-interventions, to improve data collection, and as a result, they may change the outcome of the trial. Similarly, extra data collection visits and more intensive data collection may also change the apparent outcome of the trial, the apparent effect size that's measured by the trial, as might efforts by randomized trial staff to optimize clinician and patient adherence, and also specifying protocolized care with extra investigations and co-interventions and rigidly evidence-based care for all other things. Now, when randomized trials choose outcome measures that are not directly relevant to patients and providers, the trial findings become less useful for decision makers. These biological proxies, often biomarkers rather than patient-relevant outcomes, are chosen because they're more responsive to the intervention. There is an exaggeration of meaningful effects. Or the outcomes may be very short-term rather than the longer-term outcomes, which are more relevant to patients and providers. And finally, the traditional comparators that are used in trials are very different from the situation in usual care, and therefore the findings may be difficult to apply directly to a usual care situation. So traditional trials may use a version of usual care that is lower than the usual dose or an outmoded drug. They may use an interactive placebo with blinding, which of course eliminates any belief-related mechanisms of action. And those, of course, may be active in usual care. So in some situations, blinding and placebo can reduce the apparent effect size. But as a result of these problems, traditional randomized trials may not be directly applicable to any target population setting or usual care environment, and often not even to the actual site in which the trial was conducted after the trial resources have been removed and the extra care delivery has been withdrawn once the trial is complete. Now, lacking this similarity to any usual care situation, the traditional approach to designing randomized trials may not be able to offer the kind of finding that is directly supportive for decision-making by clinicians, patients, or policymakers. Now, randomized trials are the foundation of evidence-based medicine, but we owe our understanding of the basic inadequacy of the traditional randomized trial approach for decision support to two French statisticians, Daniel Schwartz and Joseph Lelouch. Like biologists using genomics to separate what was thought to be one species into several, Schwartz and Lelouch revealed that randomized trials may be of two types, each different in purpose and in necessary design constraints from the other. A trial maker might therefore have either one of these two different purposes for doing an RCT in mind. Their randomized trial may be explanatory, aimed at studying mechanisms of action, or it may be pragmatic, focused on decision-making. A good randomized trial can be pragmatic or explanatory, or a consciously designed mixture of these for different features of the trial. Good trial design is not about making your trial maximally pragmatic or maximally explanatory. It's about aligning the design characteristics to match the intended use of the findings from your trial. So trial makers should select their main purpose in advance and design it accordingly. Explanatory randomized trials use randomization for internal validity and reduce variation as far as they can, other sources of variation as far as they can, by removing from action any other mechanisms that may affect the outcome. They achieve this by constraining the outcomes, the comparators, the analyses, and by adding extra resources to the trial. Explanatory trials clarify and make very visible what the mechanism of action under study is by constraining all other mechanisms of action. And like a highly constrained laboratory experiment, this maximizes power to refute the null hypothesis. So explanatory trials are very valuable. They add to our scientific knowledge and understanding of mechanisms of action, but they may so distort usual care that they are not able to directly contribute to decision-making about options, about the preferred option for care under usual care situations. On the other hand, pragmatic randomized trials are aiming at choosing between interventions. They're designed for both internal and external validity. They use, like explanatory trials do, randomization to eliminate confounding and non-specific factors to maximize internal validity. But they improve external validity or increase external validity by ensuring that usual care conditions prevail. The pragmatic approach is more suitable for decision-making, even though it may not neatly dissect out the mechanism of action of the intervention. But even now, most of us trial makers design randomized trials or use randomized trial information without considering the purpose for which it was intended, and thus the design features of the trial which was used to produce that information. If we don't match our design choices to our intended purpose, to our intended use of the information in the trial, we end up with what Schwartz and LaLouche called an inadequately formulated randomized trial. If our purpose is to support decision-making, we need to decide what our intention is for the trial before designing it. But by default, we design trials as if our responsibility ended with specifying randomization to attain internal validity. And so we, by default, incorporate the traditional distortions that I described. Most randomized trials are and should be aimed at supporting decision-making. As Richard Grohl, an implementation researcher, has said, evidence-based practice needs practice-based evidence, that is, evidence collected under conditions of usual care. And this accords with Schwartz and LaLouche's conclusion over half a century ago, that a pragmatic approach to trial design is often more justifiable and more useful. Alison Howie will now describe the Précis 2 tool that we developed based on the work of Schwartz and LaLouche.